Okay, how can we detect viral growth? And then the next question we will show in after a few slides, how can we quantify the viral growth? So let's imagine that you have grown a virus in one of the three flasks, so flask 1 and 2 and 3, remember, when you are using the tissue culture for cultivating the virus. So you cultivated a virus in one of the three, but you forgot to label which one, which is a very big mistake in the lab, forgetting to label the flask, or mislabeling and missing the details. So let's imagine that you did this mistake, and now the question is, which one of the three flasks contain the virus? So how can we know which of the three flasks contain this virus? The first method that we can detect the viral growth using it is the presence of the cytopathic effect. Remember, when we do the tissue culture, if the virus is specific to this type of tissue, it will do as damage for the cell and produce this inclusion bodies, which is an abnormal appearance of the cell. So one way to detect the presence of the virus, so if you wonder which of the three flasks, what you can do, you can take a sample of the cell culture, put it under the microscope and examine it. The flask in which the cells show the cytopathic effect will be the flask containing the virus. Remember, here you are examining the cells. That's why you are examining it under the normal microscope. You are not actually observing the virus itself. You are observing the damage in the cells caused by the virus. And since the host cells are large enough, they can be examined by this simple microscope. So this actually compound light microscope. Okay. Some other viruses, they cause the adsorption of red blood cells around them. So this virus here, it can cause what we call heme absorption. So the adsorption of red blood cells around it, and this can be used for the detection of the presence of a virus. But as I said, this is a property that not all viruses have. It is a property of certain viruses. So if you know that your virus is capable of heme absorption, you can use it as a method to detect the virus growth, the viral growth. The third feature you can use is the fluorescent antibody staining. If you have an antibody against this virus, you can label it with a fluorescent tag, and you can add it to the culture containing the virus. If your antibody will find the virus, it will bind to it, and since it is fluorescently tagged, you can observe it. For example, you will see these masses of cells, and they are all fluorescently glowing because of the antigen-antibody interaction. You can use this antigen-antibody interaction without the microscopy part when we move on to something called serology. So serology is the part of science which involves the use of antibody to detect the presence of antigens. And as you know, viruses, they contain antigens because they are foreign to our bodies. So our bodies produce antibodies against these viral antigens, and we can detect that by antigen-antibody interaction. They will form this type of agglutination, and you can observe it in a microplate like that or in tubes like that. Okay, so these are the methods that allow us to detect the presence of viruses. So how can we quantify viral particles? So let's say it in like a form of a scenario. If you are in the lab and you have three flasks, flask number 1, flask number 2, flask number 3. So in this flask you have added like 100 microliter of the culture containing the virus. In this one you have added 500 microliter, and in this one you have added 1000 microliter. Okay, so let's imagine that again you did the same mistake again and you forgot to label the flasks. So now you are wondering which one of the three contains the highest viral load. Is it this one or this one or this one? So without the proper labeling, without the amounts shown here, you will not be able to judge that by your naked eye. So what you can do to quantify the viral particles is using one of these methods. So you can use the Plaque Assay. If it is a bacteriophage, if it is a virus that will infect bacterial cells and cause the lysis of bacterial cells, you can detect that by observing the plaques that are produced. So here, let's say you did like duplicate of each. So you have the 3 flasks, if you remember. Here the number of plaques is the largest, and you can see that this should correspond to the highest viral load, while this should correspond to the lowest viral load, and this is something in between. Alternatively, you can use the electron microscope, and in this case you will count the number of virions. So let's imagine it is not a bacteriophage, it is an animal virus. I can use the electron microscope and count the number of fully mature viral particles using the electron microscope. Okay, we have discussed viruses that are capable of heme adsorption. In this case, we are talking about a similar feature, which is heme agglutination. Some viruses, including, for example, influenza virus, they are able to agglutinate red blood cells, causing the formation of these aggregates. They cause the red blood cells to clump together. And as we said, certain viruses do that. They are not a feature. It's not a feature for all viruses. So one way I can quantify the viral load is by observing the effect of heme agglutination. This will give me an indication about the viral loads. Or alternatively, I can use the reverse transcriptase Pcr. So reverse transcriptase, let's imagine you are examining a viral of a virus with RNA genome. So you will use the reverse transcriptase to convert this RNA into the complementary DNA. And once we have DNA, you can use the real-time PCR in order to compare the viral loads that you have in the different specimens.